So I am delighted to welcome our next speaker, and due to internet issues, all of our other speakers have kindly pre-recorded their session. So our next speaker is Damilare Ayokunle from Bowen University in Nigeria, and Damilare is a PhD researcher, and she's going to be talking about isolating bacteria of the straw-coloured fruit bat. So again, a topical one following on from Dina.
Hi everyone, I'm Ayokunle Damilare from Bowen University in Nigeria, and I'm happy to be here. I want to congratulate Bats Without Borders on this first Women in Bat Science Indaba, and I'm happy to be here. Thank you for the opportunity to come talk about one of our projects on Indaba bats, that's the isolation and molecular characterization of bacteria from their guano, that is their dropping, their fecal matter. And here are the research team members that will work together. This project is a very interesting one because I had to go source for microbiologists to come together for us to work something out on their fecal matter. Idolon are known as the palm bats, and we have two species of that, and the commonest species we have is the straw-coloured fruit bat, which we have in abundance here in Bowen. Bats are known to be reservoirs for a variety of pathogens, as we all know, viruses, fungi, and bacteria. And also in communities where we have a very high number of them, like Bowen University, for example, we discover that their roosts, in a large roost, their droppings can accumulate over time. Their droppings are the fecal matter, their fecal matter, that's also known as guanos. So we discover that over a long period of time it can accumulate and then dig several meters deep, hereby forming complexes and interactions with organisms in a habitat with low light or even oxygen. So as they decay, the nutrient composition of that particular area that's under the roost changes, and also the bacterial community deep within the pile. We start to observe them, and they become, the bacterial community becomes increasingly, obvious. And then these bat piles have not been fully characterized yet. We have some researchers that have talked about how we can get a lot of things from the bat guanos, so we just decided that we can also go into it. And as we all know, bats, they rest during the day or forage at night from dusk till dawn and all of that. Same applies here also. You see them at night from around 6 p.m. they start to move. They start to leave the campus to go and feed. And aside their noise, we discover that their fecal matter gives off offensive smell and odors, and pollution of the hair and everything. But then we researchers just decided that there should be something that we can get from their bat guanos, and that was why we went ahead. So here the objectives include mapping out roosting location of straw-coloured fruit bats on the campus, and also to isolate bacteria present in the bat guano, then also to identify the bacteria that have been isolated using series of analysis. Let me just say this work is just like a fragment of a bigger project, and it's still ongoing. So this is just like a progress report I'm going to give them. That is going to just tell us about what we've been up to and what we've been doing as regards Indaba in Bowen University. This study area that Bowen University is located at, the geographical details given, and the university is the foremost Christian Nigerian university that is owned and operated by the Nigerian Baptist Convention, and it is located in Iwo, and Iwo is known to have a population of over 250,000. The population is over 250,000 as at 2022. So I try to give a picture of what a bat roost, one of the bat roosts in Bowen looks like, and also how the droppings how the droppings appear on. This is a cemented platform, just to get the contrast of colors. Where we have the orange color, we have the dark and brown color. So the dark and brownish pellets or droppings are the ejectile. These are what over time, if they are not cleaned, so they clean this platform on a daily basis. So in a place where you have the bathrooms where it's not cemented, or not, they don't have access to cleaning every day, that is where you can have the pile of the bath dropping, the guano. So one of the objectives is to map out bath rust, and we did that here. And then after that, the samples were collected. Fresh bath guano pellets were collected from 5 selected sites within Bowen University. And how was this done? We made use of serialized black polythene bags, and we placed them under bath roosts after they might have gone to feed, because we didn't want any contamination. And as early as 6 a.m. the next day, we go to recover the polythene bag, which would have been filled with droppings and of what we need. So we take that back into the laboratory and then for further analysis. So this was gotten from 5 selected sites, and a total of 5 fresh guano pellets were collected with the polythene of sufficient quality of fresh samples free from contamination. Here is giving the information about the collection, the roosts, the names of the tree species used, the common name, the geographical location, and every other thing we need to know about the trees, the bat roost that we got our samples from. So in the laboratory, also we had a series of experiment and analysis, like experimental analysis. This is where we take 1 gram of the sample and mix with we mix that with water to get a homogeneous mixture, after which the serial dilution occur. We undergo serial dilution. The pop plate method was used here, and then we used nutrient agar for the media growth for 24 hours, after which the density of the growth was calculated. Then when we saw positive growth, we had to go further and purify using the quadrant streak to get better and more results. After that, we have to do the biochemical test, series of biochemical tests, just to go further into our analysis to get perfect results. Then after that, we had this organism count just before the molecular characterization, where we had majority of them being the Staphylococcus intermediate, that is. Then we had the Staphylococcus aureus and the other ones that were. So from that sample, we had to pull out the most occurring ones for the molecular characterization. So at the end of the day, we got another beautiful organism as a result of the molecular characterization. And here is the phylogenetic tree analysis of our samples. Because work is still ongoing, I may not be able to talk so much on our results just yet, but there is the study validated the fact that, number one, bat guano can be assessed for bacterial colonies, just as we have. And the 3 general we found so far are of research, industrial, and commercial potential. That means further work can still be done on them. The second milestone is that bat guano in Bowen University collected are mainly a single bacterial group, that is gram-positive. Now we have studies that have recorded both gram-positive and gram-negative group of bacteria gotten from bat guano. That is actually what we set out for, but we didn't see gram-negative bacteria. So also another interesting thing about our results is that the bacteria that were isolated are not pathogenic. They are not pathogenic, and we still have to go, we still have to go in more and do a lot of other study to know why this is what we have. But according to our study, this is what was obtainable, and we made do with them. Just like I said that there are a lot of work that still be ongoing, I just decided to have some beautiful charts we've had on the field, telling us how work is done here in Bowen, making use of our beautiful bats and we make sure we release them back after we get our samples from them, because no bat has to get injured that much for now. And this is what we had, and it's been a beautiful ride, and I want to thank you for listening.
Thank you very much. That was, I'm just double checking Damilare is online for questions. Here we go.
Yes, I'm
sorry, there's quite a few people. I saw you popped in earlier at the start, and then here we go. Thank you very, very much for your talk. And I know that actually, so Damilare is also one of our committee members as well, and I'm sure would appreciate, as many of our PhD researchers and master students can often be quite isolated in, they're sometimes the only people at the universities working on bats. I know you have some other collaborators, so that is great that you have some support, and also hopefully within this network. So I just wanted to kind of start off with a question of, what are the challenges you think for your early career, I suppose, getting started on your research with Eidolon? How have you found that particular journey?
Thank you very much, Rachel. It's been very challenging, and I think it's taken a longer period for me because, number one, I don't have much data to work with for my doctoral degree. It's like I'm starting from the scratch, going to the field. And being a female, I have to, employ a lot of masculine hand on ground. By the time you're going to the field, you have to sleep over in the forest and all of that. So I've been getting at least it's getting better over the years. But my first 2-3 years were very, very challenging. And then I would just say I didn't really get as much help as I would, but I knew, of course, I knew what I was going into wildlife and bats and all of that. So I was much prepared for it.
Great. And so, I mean, obviously with your, it's still kind of quite early days in your research, but have you found actually the capture to actually get enough samples? Have you found any challenges? So I know you're obviously collecting guano samples, but also taking other morphometric data, but have you found it actually difficult catching enough individuals? And have you, and I suppose, throughout the seasons as well, so what kind of challenges, but also I suppose, exciting fieldwork have you found at the start?
Okay, so the challenging part was trapping Eidolon bats. It was, okay, so we started out using mist nets, and we discovered that we don't really get them. Okay, my first night here, the first night of trapping, I got nothing. I stayed up all night and caught nothing. I discovered that they either escape or they go over the height of my pole, and then so I had to improvise for other means to trap them and get them in, a sizable number to work with. Then we've been trying to publish this, but let me just say we've been working on some bat flies. The work is a very, very broad one, just as I said, different aspects and sectors, but the parasitology aspect, where we're looking at some parasites that bat flies have. These bat flies are those flies you see on Eidolon earworm, maybe at the very earliest hour of trapping them before they stay too long when sampling. So we have these bat flies, and then we discovered that there is actually they have parasites in them. So that's just like hyperparasitism. That's the word for it. So we try to get a species that has not been recorded before. So we've already submitted to the gene bank, and they acknowledged it, and we're trying to put it together to release as a publication. And that's a major milestone for parasitology that we've gotten. Every other aspect has been very interesting, and it's been a joyful ride, as I said.
Great. So yeah, I can imagine, and I know, also with people looking at sort of parasites have struggled actually with, kind of keys to actually key out those species. So that must also kind of sometimes be a little bit tricky. So we have a question from Natalie saying, have you considered that you selected for only bacteria that could be cultured in a medium of your choice, and that you can produce colonies within 24 hours. In other words, bacteria that are non-cultured or slow-growing could not be detected potentially. Will this unbias the deep sequencing, not providing more complete picture of the bacteria present in your samples?
Okay, thank you very much for this question. I'll take it as a suggestion also, because another challenge I'm having is funding. So for us to be able to do a very, very in-depth molecular study, we require a lot of funding, because we require much more sample size. So this was just like a preliminary study to know, okay, what others have been getting in Eidolon ovum, can we obtain that here in our guanos also? And since we got a positive result, no, the second step of this study, the study I presented actually is to check for the antibiotic resistance activity of the bacteria we got first. Then we can also look into this suggestion. So that's why I said this is a beautiful suggestion. Thank you very much.
Excellent. Well, thank you very, very much for your time, and it's really exciting to hear more of your research. And we just want to thank you very much. And also, if you want to carry on the conversation in the chat and answer any other questions, feel free to continue this conversation throughout today.
Thank you. Thank you so much.
Very much.